so I'll be talking about deep learning and deep time. So for this group, I don't think I need to belabor the point about how amazing the diversity of life on Earth is, the morphological disparity that exists. But of course, as we've kind of heard from the earlier talks today, this amazing diversity that we see on Earth today, which we can barely get a handle on, is just a teeny tiny sliver of the diversity of life that has existed on Earth. So once we bring this time dimension in, things get a bit more complicated. So for most of life history on Earth, morphology is what we really have to investigate this diversity. This group, I think, understands the importance of morphology. I often give this version of this talk to departments that are filled with geneticists who really need a reminder about how important morphology is. And when it comes to life in the past, so actually at Stockholm University, there's a very big, famous group that works on ancient DNA, the Center for Paleogenetics. You may have seen some of their work. They do a lot of work on mammoths, for instance. And ancient DNA has really, grown by leaps and bounds in the last few years. But the oldest ancient DNA that we have sequenced so far is about 2.4 million years old. Now, if we assume that the current best estimate for the age of life on Earth is correct, which is about 3.7 billion years, this means that our ancient DNA covers about 0.06% of life history on Earth. And that's assuming that we have, perfect ancient DNA sampling in this period of time, which we absolutely do not. Ancient DNA requires really amazing preservation in order to actually find it, which is why all this work that you see, most of it's being done on stuff that's been stuck in the permafrost since the last ice age. Right, so if we go back any further, morphology is really where it's at. So my research is focused on, so I'm really interested in understanding macroevolutionary patterns and understanding the evolution of communities through time and space. And so to do this type of work, you need a lot of data. And traditional ways of collecting this data, of course, take a lot of time, especially when you're working with really, really teeny tiny things like foraminifera. So what I'm really focused on now is trying to use these high throughput methods, use these AI methods, deep learning, to try and make this process faster so that we can do the data extraction side of things as quickly as possible, so that we can instead focus more of our time on the actual data analysis, answering questions side of things. So as I mentioned yesterday, the organisms that I work the most on are called planktonic foraminifera, and today you'll finally get to learn what these things are. So planktonic foraminifera are single-celled amoeboid protists, so they are a single cell. They're split into, so there are planktonic foraminifera, which is what I work on primarily, and there are benthic foraminifera. So planktonics live in like the upper surface waters of the ocean. All over the world, benthics live in the sediments on the ocean floor. And there are many, many thousands of species of modern benthic foraminifera, but planktonic foraminifera, there are only about 50 extant species. And I say about because, as I mentioned yesterday, there's a lot of debate about where these species boundaries actually are. So depending on who you ask, but the number is around 50. So the important thing about these guys is they construct this external shell or test out of calcium carbonate. And this is really important because what it means is that these guys preserve really easily in the fossil record. So they're all over the world. They float in the ocean, on the surface waters, and they die, and these shells sink down into the bottom sediments. They get buried, and they get fossilized. So they've been around, planktonics have been around since the Jurassic, about 200 million years ago. And here's a phylogenetic tree. You can kind of orient yourself. So we are over here. So animals and fungi are in Opisthocanta over here. So plants and green algae are over here in Chloroplastida, and foraminifera over here in the SAR group in Rhizaria, along with radiolarians, if you've ever heard of them, another group of planktonic organisms. So forams have this really incredible fossil record. They're very easily preserved. They're all over the place. And they're so really one of the most complete fossil records of any type of organism in the world. And because they have these calcium carbonate shells, what we can actually do is use isotopes to reconstruct climate in the past using these organisms. So these organisms, even though you may never have heard of them before, are really, really important for paleoclimate reconstruction. And a lot of our understanding of climate in the past is based on these organisms. And so they're also really important for studies dealing with climate change now, because we can use them to look at how climate evolved in the past during events of warming and so on, and apply those lessons to what's going on today. So these isotopes can be used, so oxygen isotopes can be used as a proxy record for sea surface temperature. These carbon records can be used as a proxy of oceanic carbon cycling and so on.
Sorry, Allison, is it up or down? Go back to the slide. Is sea surface temperature up or down for those two different proxies? Is the value, when the value goes up, does the sea temperature go up, or does the value go down, sea temperature go up? On the y-axis.
You have a right line.
So oxygen is up here.
I'm sorry, 'cause I'm not saying.
So here's oxygen. This is lower
No, I get it now. I
value.
just didn't see
Yeah.
it was a right-hand access. I
Yes.
wasn't expecting that. Thank you.
Okay. Right. So another important thing about these organisms is that their morphology also correlates to some degree with their environmental surroundings. And also their ecology. So this on the left here is a plot showing average abundance as temperature changes for some different species of planktonic foraminifera. Also, there are things like the size of the test is dependent to some degree on sea surface temperature. Things like coiling direction can also be dependent on sea surface temperature to some degree. So that makes them really interesting for sort of correlating between these environmental variables and their morphology. So I imagine most of you aren't so familiar with these guys, so just some very basic stuff about how we collect these samples. As I say, they sink down to the ocean sediments. What we do is called coring. So basically you have this hollow metal thing that you drill down into the ocean sediments, and then you pull it up, and you get this long core of sediment that you can then go in and date the different layers and find their foraminifera and so on. Yeah, so this is more for collecting live ones. There are different types of traps that you go and collect, but that's not really relevant for what I'm talking about today. What I'm talking about today is all to do with samples taken from these coring expeditions. So we want to build these really large data sets, and traditionally we kind of take like a sample of 300 forams, and we can measure them and identify them to look at the communities. But this, if you have a few, it's really easy for someone to sit there and identify the species and measure them using calipers or what have you, image J by hand. But if you scale that up, then of course nobody wants to do that, right? So a lot of what I do is just trying to deal with what do we do when we have all these specimens using automation and machine learning. So I brought up automorph yesterday in my talk. So just a reminder, so this is a program that we wrote to take images that have lots of microfossils on them, but I mean, you can use it for anything you can take pictures of, basically. And it will automatically find where all the objects are, and then also take these morphometric measurements. So a bit more detail, but we saw this yesterday as well. Here's how it does it using these traditional image processing algorithms. So once it gets these outlines, which it does by doing this whole process, once it gets these outlines, you can very easily get out these measurements, right? You can get coordinates, you want to do outline analysis, major minor axis length, area, perimeter, aspect ratio. If you have these Z-stacked images, you can also get estimates of surface area and volume for each of these objects quickly and automatically. And this is just from the paper describing automorph, showing where we basically measured a bunch of things by hand and measured them by using automorph to prove that it works. But also what we showed down here is, with reproducibility. With automorph, you run it, you get the same numbers when you run it. This is when you compare it to hand measurements in image J, the traditional way of doing things. You see there's a lot more spread, so people don't get the same measurements every time. So that's another benefit of automating these kinds of things. And if you do that, you can, of course, very quickly do traditional things like look at the distribution of size in a population. You can build these morphospaces using the morphometric data that you get. And some examples of other things that people have done using automorph. So this is a project that Sara Kahanamoku led. So when she was a PhD student at Berkeley, so she's now actually at University of Hawaii in Manoa. But she imaged about 12,000 of these limpets from the coast of from the west coast of the United States and up through Canada, and she used automorph to be able to segment out all these individuals and look at the 3D and 2D morphometric information. So not limited to, you know, micro scale stuff. You can do it. We did have to like rig a different kind of camera setup, because the problem with bigger things is that as you move the camera, the object moves in the field of vision. So then we had to deal with that. There's an option in automorph to kind of correct for that sort of thing. This is another example. So this is Elizabeth Seibert, who is now at the Woods Hole Oceanographic Institute, and she used automorph to look at ichthyoliths. So these are fossil fish teeth. And she was able to do this to reconstruct morphospace occupation across the KPG max extinction, which I'll talk about more later. But she was basically able to see that there were two pulses of morphological diversification in the aftermath of this extinction event by being able to measure a ton of fish teeth very quickly and build these morphospaces. So now we'll talk about what I did with automorph, the reason that I put together this program, which was to build this endless forams most beautiful database, which I talked a little bit about yesterday. So here again is the pipeline. I won't belabor this since I went through it. You know, you have your samples, you image them, you run them through automorph, you get your objects. These samples come from, so some from the Yale Peabody Museum of Natural History collection. So here we sample 34 Atlantic core tops, mostly from the northern Atlantic. You can see them here. And we basically imaged everything that was in those core top samples after washing and pre-processing and so on. And yesterday I talked about how we did coarse classification to separate out all the planktonic foraminifera from the other things. And we ended up at the end with about 61,000 planktonic foraminifera across these sites. In addition, we also had samples from the Henry A. Buckley collection from the Natural History Museum in London. So these were forams collected by Henry A. Buckley, who collected these forams, prepped them, and put them on these slides and identified them to the species level. So it was nice that we already had species identifications for all these, although we did go back, as I said, you should have people check. So we did have 2 people go back and independently check through all of those species identifications to make sure that they all agreed on what it was. And these samples are from all over the world. So to collect these species identifications for the Yale part of the database, we had a team of about 20 taxonomic experts who basically just sent an email out and asked to all the taxonomic experts we knew and said, hey, do you want to join our project? And these were the people who joined up. And then we collected all these identifications, as I mentioned yesterday. So in the end, as I said, we had the quadruplicate classification for every object, and we only kept the ones where at least 3 out of 4 people agreed on what it was. So this is what we call high quality classifications in our data set, and we threw out everything else. So in the end, we had about 24,000 individuals with these high quality labels from the Yale data set, which we combined with these 10,000 individuals from the NHM to get our data set of 36,000 or so planktonic foraminifera, which represented 34 species. And it's quite uneven, as you can see. There's a long tail. But for 7 of these species, we had more than 1,000 images for these species. So that's quite cool, because what I think is one of the most interesting aspects of this data set is that it really shows you the amount of intraspecific variation that exists within each of these species concepts. So these are some example plates from our more some of the species where we have more individuals, and you can see, within each of these, there's a lot of variation within this one species concept. And so we really wanted endless forams, not only we built it to train, this machine learning model, but we really also want it to be a resource for people to sort of learn the taxonomy of these types of organisms and what that variation is interspecifically and intraspecifically. So if you go to endlessforams.org, you can download the data set, but what you can also do is you'll see there's a quiz function up here. So if you click on that, what happens is that it will bring up random images from the data set, and then you can there's a list of species, and you can choose which one you think it is. And then It will tell you if you're right or wrong, and you can also show answer if you have no idea. And in that way, kind of train yourself to recognize some species. So I actually am really bad at this. I do more of the method stuff. I'm not a taxonomist of planktonic foraminifera, but now I can recognize a few of them because I've done that a few times. So if you're interested, you can go try and do that. So we built this data set to train this supervised machine learning model. I'm assuming that after all these talks, you guys have an idea of how this works. It's a cartoon where you have your training set of images and labels, you throw it into your model, and then hopefully, if you train it well, you get a model that can look at entirely new pictures and classify what's in those pictures. So we did a standard 80-20 split, and then we trained this model. So we tried a few different architectures, but we ended up going with the simplest architecture that we tried, which is the VGG16 architecture, because that gave us the best results. So going back to what Ben said yesterday or the day before about, just try the off-the-shelf models. They can do a lot more than maybe you expect. And we had a model accuracy of about 87%, which if you recall what I was talking yesterday about our human accuracies, they had an average accuracy of 71%. So you can compare those two and see that our model is doing better than the humans, even better than our best performing human, which is quite cool, I think. So we have some collaborators at the University of Bristol who also took our data set and applied a different model to it. This is a deep metric learning model in which they pushed the accuracy up to 92%. So that's, yeah, that's the current state of this model, I guess. So here is the confusion matrix from our original endless forums paper. This is a confusion matrix for the machine. But because we collected those quadruplicate classifications from the human side, what we can also do is build a confusion matrix for the humans. So what we're basically doing here is we're going to all those instances where only 3 out of 4 people agreed. We look at what did that 4th person say they thought this thing was. And then from that, we built this human confusion matrix. And then what we can then do is compare, when the machine gets stuff wrong, what is it getting it wrong? What is it confusing things for? Compare that to what humans are confusing things for. So one of the things that we found quite strong evidence for was if you look at this here. So basically, this plot here is looking at, so for each species in our data set, the y-axis is the number of samples we have for that particular species. The x-axis is the number of misidentifications attributed to that species. So basically, when someone gets something wrong, they say, this thing is not rubor, but they say it is rubor. That's one of those misidentifications for that species. And what you see here is that for humans, there appears to be a very strong correlation between the number of samples in the data set of a particular species and the number of times they wrongly attribute something to that species. So this is, we call this basically like a rarity bias, where humans will tend to classify things that are rare as something that is more common in the data set. Particularly since, when we served up these images to the classifiers, they had access to the metadata associated with each image. So they know where it's from, they know which gives them information. They know where it's from, they know the size, various things, which gives them some information, some context with which they're probably making some of their decisions. So if they know, for instance, from their experience, this site has a lot of rubor, they might see something that is very rare in that site, but looks kind of like rubor, and just be like, okay, that's probably rubor. So we think that's what's going on here. This is the equivalent plot for the machine performance, and you can see there is some correlation, but not anything nearly as strong as what's going on with the humans. Another thing that we noticed was that human mistakes tend to be phylogenetically conservative, and what I mean by that is that humans will tend to, if they get something wrong, they'll get it to the same genus, they'll just call it a different species. Whereas with the machines, don't necessarily do that, but instead they do what I call sort of historical mistakes. And what I mean by this is that, so there was sort of a revolution in planktonic foram taxonomy when the SEM imaging technique was introduced to studying planktonic foraminifera. So people who worked before there was SEM, they had some idea of the taxonomy of these organisms. Then SEM came in and they looked at these, much nicer images where you can look at the texture of the shell, you can look at much finer grained details. And based on that, a lot of the taxonomy was revised. So the taxonomy we use now is based in part on this SEM data that we now have access to. But the machine model that we've trained here is only using light microscopy images. So actually we find that a lot of the mistakes that the machine is making are basically sort of reprising what humans thought was going on in this group before we had SEM, which we thought was kind of interesting. So, yeah. So now we have this trained model that can identify species of planktonic foraminifera with quite high accuracy. And now we wanted to sort of test out sort of the dream situation, right? Which is, we have these models, yes, but what we really want to do with them is to be able to use them to predict species labels without us humans having to go in there and do that. So we have our endless forams data set. You'll see that the number of specimens here is a bit lower because we downsampled to just the ones that had, the sites that had very good preservation. But the rest of the Yale data set was still there. We didn't have labels for the rest of those images, and that includes all the images that the humans couldn't agree on a species label for. So these come from the same sites. The selection of individuals that made it into here was random, the images that we put onto the Zooniverse database for people to identify. So what we basically have here is two random samples from the same underlying collection of sites, except one has completely human labeled species labels, and then one we just applied our machine learning model, so now we've got this independent data set that has labels that are completely derived from our machine learning model. And what we wanted to do was basically take these two, run the same set of analyses on them, compare them, and see are there any differences. How feasible is it to actually do everything by machine for the data collection side. So we ran a series of analyses characterizing community ecology. I'm not going to go through all of these, but this is what we looked at. So just to highlight a few things. So here we've got species richness. So in all these plots, so red is the human data set, blue is the machine data set, purple is combined. And what you see here is, what you would expect, this traditional pattern where you have, lower species richness at the high latitudes and higher species richness at the equator. But what you'll see is that there's an offset between the human data set and the machine data set. And we think this is another illustration of this rarity bias that I was talking to you about how humans just kind of miss out on rare species because they kind of preferentially assign them to more common species. And we think we can see this bearing out in the species richness curves. We see something similar in the body size. So here we have area, here we have volume. These are all extracted from automorph. What you see here is, again, the traditional pattern. You have smaller body sizes at the poles, larger in the equatorial regions. But the offset is kind of inversed here, right? So the machine learning curve is, the individuals tend to be smaller than the ones in the human data set. And this is likely because with planktonic foraminifera, it becomes much, much more difficult to identify them the smaller they get. So probably what's happening is when people were identifying things, we weren't getting, high quality labels for all the small guys because people couldn't agree on what it was or so on. And so those individuals basically kind of got shunted into this unlabeled data set, which we are now labeling using the machine learning model. So you can actually see this in the sizes, relative sizes of the organisms in the two data sets. Another pattern that we notice, so this is a plot looking at the relative abundance for all the sites that we have. So within each of these sites, there's the combined data set, the human data set, and the machine data set. So it's not so easy to see here, but something we noticed was that in sites where, so let's zoom in on one of these. So in sites that are relatively dominated by one or two species, what we find is that humans tend to assign way more species into these one or two dominant species than the machine does. So in contrast, this is a more evenly distributed site. In contrast, when it's more even, there tend to be more individuals within each species in the machine data set than the human data set, which is what you would expect if there were no bias, because if you remember, there are about 5,000 more individuals in the machine data set than there are in the human data set. But we don't actually see this when a site is dominated by one or two individuals. And we think, again, this is the rarity bias coming into play, where, if you know that a site is dominated by these one or two species, then you're going to preferentially put things into one of those categories. This is just another illustration of this. So this is looking at the Jacquard dissimilarity index between two different communities. So here's for the human data set, machine combined. And here we have the difference in the Jacquard's dissimilarity measure between the human and the machine. And just highlighting one of these here, but all the strong differences that we see between the two data sets, when you actually go in and look at what is the composition of these communities, you'll find that the machine data set often contains rare species that don't show up in the human data set. So we think there's a lot of evidence for this rarity bias in humans from all these lines. Okay, so now I'll talk about what I'm working on now at Stockholm University. So this is a project that's focused on the KPG mass extinction, which is the Cretaceous Paleogene mass extinction. Even if you don't know this name, you absolutely know what this is, because this is when the boli collided with the Earth 66 million years ago and the dinosaurs all went extinct. But it isn't just the dinosaurs that went extinct. So planktonic foraminifera also went well, not completely extinct. Well, actually, it depends on who you ask, but you can ask me about that later if you're interested. But planktonic foraminifera suffered very high losses at this time. So about 90% of them went extinct. And this is important because planktonic foraminifera are a very important part of the biological pump. And the biological pump is basically the system whereby carbon in the atmosphere is basically brought into deep ocean sediments as a carbon sink. So planktonic foraminifera and other organisms, they take, carbon goes into the ocean via, atmosphere-ocean exchange. Organisms like planktonic foraminifera take this carbon, use it to do things like build their little shells out of calcium carbonate, and then when they die, this stuff sinks down to the ocean, gets buried, and so that's how one of the ways carbon gets cycled between the atmosphere and the deep ocean. So when 90% of planktonic foraminifera went extinct at the boundary, the biological pump collapsed. So this was obviously bad. And other evidence has shown that the biological pump function seems to go back to how it was operating before the extinction event by about 2 million years after the boundary. However, planktonic foram diversity does not recover to what it was before by 2 million years after the boundary. Right, so we've got the situation where in the Cretaceous we've got this really diverse, really disparate planktonic foram communities, and then we have our extinction event. We end up with 3 surviving species, and then they recover, but they don't get back to what was going on here, and certainly not by 2 million years after the event. So the question of this project is basically, what's going on morphologically on a community level, and can we correlate that those patterns with what's going on environmentally and with what we know about how temperature changes, how the environment changes, how the biological pump recovers in the aftermath. So the samples for this project all come from the Walvis Ridge. So this is a very famous planktonic foram KPG site. This was one of the sites. There's these big ocean drilling projects where they go out there and drill all these cores. So it's from one of those really big initiatives. And people the KPG is obviously a very popular time to study. People have looked at, the isotopes, looked at the environmental proxies across this period. So we have really good environmental proxy data for this time period, which we can then compare to this high-resolution morphological record that I'm building. So the basic idea is that, this part is the same as before, but so we have our samples, we image them, so we do what we end up with is our Z-stacked images, 3D images, and then the 2D images. So the 2D side will push this down into the computer vision part of this project, which is to use like mask-R-CNN, these segmentation models, to identify where all the objects are and to also distinguish between are they damaged, are they complete, and so on. Which we get, then we get these binary masks of where all the objects are. 3D side will do processing to basically, the microscope that I'm using will output a CSV file of all the 3D information, and then you just have to kind of match it with your objects and cut out that 3D information and associate it with each object, and then do the morphological data extraction using automorph and other tools. So this is the microscope that I'm using. So this is an automated light microscope. What you basically do is you can set a slide on there, you tell it where the borders are, and then you let it run and let it do its thing. And then you end up with something like this. This is one of these slide images. This is a close-up of some of these individuals. So the total time period that we're looking at is a period of about 2.5 million years. So we're going from about 500,000 years before the boundary, so we can get a sense of what the community was like before the extinction event. And then we go through the boundary and up to 2 million years, which is when the biological pump has recovered. So we're really focusing on this recovery period of the biological pump and comparing that to the morphological evolution. So everything is split up into 4 size fractions. So you can see an example here. So this is 150 microns, 125, 90, everything below 90 microns. And in total, I have 772 slides across 193 time slices. And there's an average time of about 13,000 years between each time slice. The important thing about this project that is distinct from the Endless Forams project is that it's orientation free. And what I mean by that is that the Endless Forams project, we had people sort of painstakingly pick forams and arrange them in a certain, it's called umbilical side up. Here, that takes a lot of time. I don't want to mess with that. So here what I'm doing is just pouring the sample on the slide and imaging it and hoping that the model will be able to deal with that. And everything's at 200x magnification. This plot down here is just, if you're interested in using this microscope, this is just, I plotted how long it takes to image one of these images depending on the range of, the Z range that you're imaging across. But on average, it takes somewhere from 1.5 hours to 2 hours to image every one of these slides. So this is just focusing in on one particular sample in this data set. So these are the number of objects that I counted. Well, I didn't count by hand. The number of objects that were counted using automorph for this one sample. So you can get an idea of how many objects we're talking about here. So this particular sample has about 58,000 individuals. If we assume it's more or less the same for all the time slices, then we have in total about 11 million objects across all these time slices that we're dealing with here. So orientation free is great in that it makes it so you don't have to do all this pre-processing, but it does mean you have to identify where certain types of objects are. So here is a damaged foram, here is a fragment of a foram, so we need to have our model be able to go in there and find these things. So for that we're using mask-R-CNN, which people have talked about already, so I won't talk so much about how it works. But basically it's a segmentation model that you can identify where the objects are in the image and also have it predict a class. So it does this by proposing, so the R stands for region-based, and it basically proposes all these different regions and calculates a score, and then to figure out what it is and where it is. So the type of training data that you need is basically a pixel mask. So here, for example, is part of an image, and this is the training data that I'm collecting for all these images. So is a complete damage, fragmented, or occluded. So to help with the labeling, I'm doing model-assisted labeling. So this is like what was mentioned before about having these loops to sort of iterate over and over, where you basically, you label some, you train the model, you have the model predict on other images, and then you go back in there and correct their predictions, and then you feed that back in to train the model and so on. So the annotations that I have so far are about 20,000 forams, about 3,000 damaged, 4,000 fragmented, and 6,000 occluded. And then what I've run so far is do the standard 80-20 split just to see what's going on here. So the performance of the model right now is not super great. So I get about 50% of all my objects. And this problem is entirely to do with the classification side of things. So segmentation it has no problem with. If I go in there and I mess with the threshold that it's using for these objects, I can see that for the objects that it's not getting, it's getting it, but it can't decide whether it's complete or damaged or fragmented or whatever. And so I think moving forward, one thing that I really need to do is I need to follow my own advice and have other people look at these labels, because right now these labels are only based on what I have done. So yeah, increase the quality of the labels by having, I'm in the process of recruiting people right now to help me with this. And also it's just kind of a hard problem, right? Because the distance between these classes is really quite small. So if you imagine the distance between a cat and a car is quite large, but the difference between a foram that's complete and a foram that looks complete but it has a tiny hole in it, I mean, that's a much trickier problem. And also, some of these things, they have, foraminifera means like hole bearers, and they have these little holes or apertures, and some of them have secondary apertures. So most of them only have this one on the outside, but some of them have secondary ones, and some of these holes might not actually be damaged holes, they might actually be holes that are secondary apertures. So there are things like that that make this problem difficult. So if anybody has any experience working with classes that are so close together, I'd really love to talk about potential solutions that you might have. But for the rest of this, we can talk about the rest of the pipeline. So what's going to happen once the model is performing really well. So we go back to this particular sample that I showed you before, and what happens is that once we have, this is from one of my hand annotations, which is why I have all this, everything nicely marked out. But ideally, once the model is performing really well, what it will do is spit out these masks, which we can separate out into each different class of object we're looking at. And then from here, we can also get, from automorph, we can get our 2D measurements for every single object automatically. And then the microscope spits out, as I said, this CSV file with all the 3D information, which then I've written a little module to automorph that will basically go in there, and you have this is one of these slides plotted out. What you can do is then just say, okay, automorph, where are all the objects? Here's one, here's the part of the 3D CSV file that is associated with this object, cut it out, save it, there it is. So this is from that site that I showed you. These are measurements that I did with automorph and plotted out, some of these morphometric measurements: major axis length, minor axis length, area, and perimeter, separated by size fraction. And it takes about 6 minutes to measure 672 objects for this particular run. That will, of course, depend on the size of your objects and your resolution and so on. But that's just an idea of how you can get a lot of measurements in very little time. And then the next step would be to look at morphospace occupation. So this is just the first 79 outlines from the 150 micron size fraction of that site that I pulled out from automorph's results that you can see here. So that's it, and happy to take any questions.